So I thought we'd go and talk a little bit more about cross-linking now. So last lecture, I briefly mentioned the fact that for collagen fibrils down here to gain tensile strength, the individual molecules need to be cross-linked to one another so that there's some resistance or increased resistance to molecular sliding. So let's just look at a tiny bit of the chemistry involved in collagen cross-linking. So just to give you a bit of a taste at how this is done, or a bit of an idea that how this is done. So cross-linking between collagen molecules always occurs due to the joining or the enzymatic cross-linking of collagen molecules always occurs due to covalent bonding between side chains of lysine residues. One lysine residue that is in the non-helical telopeptide, what's called the telopeptide region of the collagen molecule right at the end, and that joins to a lysine side chain on the helical portion of an adjacent collagen molecule. And there are a couple of steps involved in this. So first of all, the lysine side chain needs to be acted on by an enzyme, lysyl hydroxylase, that adds a hydroxyl group here. And this hydroxylation of the lysine side chain, this can either happen to the lysine side chain that's in the telopeptide region or the lysine side chain that's in the triple helical region. I have it drawn here on the telopeptide region, but it can also happen at the triple helical region too. And depending on which of these side chains becomes hydroxylated, it controls the resulting chemical structure of the cross-link that's formed. So after hydroxylation, then another enzyme, lysyl oxidase, converts that side chain into an aldehyde, which then becomes reactive and can react with the other lysine side chain, forming a cross-link. When these crosslinks initially form, there's two collagen molecules that they join, and they're called immature crosslinks because they're the ones that form first during tissue formation. They're also referred to as divalent because they join two collagen molecules to one another. As we age over time, these divalent crosslinks, right, here's the divalent crosslink again, spontaneously react with other crosslinks or other lysine side chains to form trivalent crosslinks that link three collagen molecules together. Okay, so you get a bit of a change in cross-linking over time as our tissues age. And then there's also non-enzymatic crosslinks that occur as well, more haphazardly and in a less controlled fashion than these enzymatically controlled crosslinks. And that's driven by the reaction of sugars in the interstitial fluid within tendons or other collagenous tissues, the reaction between sugars and amino acid side chains as well. Right? So you can get additional sugar-derived crosslinks too, and those are called advanced glycation end product crosslinks. So we know that these crosslinks are important to the mechanics of collagen, and we can see that from a couple of different areas. So here's a fun one too, right? There were people that used to go on long sea voyages that would end up looking like this guy here, and this doesn't so much happen anymore. Of course, this is a picture of somebody who is suffering from scurvy, or somebody at Halloween dressed up to be looking like somebody who's suffering from scurvy. And scurvy, of course, is a disease that's caused by lack of vitamin C in the diet. So long sea voyages, no access to fresh fruits and vegetables. Scurvy was very common. So what does vitamin C have to do with collagen cross-linking? Well, it turns out that vitamin C is a necessary cofactor for this enzyme lysyl hydroxylase. So this reaction here, the hydroxylation of this lysine side chain, can't happen without vitamin C. And so collagen, then, new collagen that's laid down in the absence of vitamin C, can't be properly cross-linked and ends up being very weak. And this shows up in scurvy as lesions in the collagenous tissues that are most rapidly turned over in the body. So the periodontal ligaments, for example, that hold your teeth in place, right? The collagen is turned over very, very quickly in those. So if you're lacking vitamin C in your diet, the new collagen in the periodontal ligament isn't properly cross-linked and your teeth start falling out. Skin is also another collagenous tissue where the collagen is turned over very quickly. So you end up with skin lesions as a result of scurvy in the absence of vitamin C. So one sort of historical and fun way of showing that the importance of cross-linking to collagen mechanics. Fun unless you're the person with scurvy. Another way that you can show this is by what's called induced Lathricism. So there's a compound in this plant here that looks like snow peas, but it's not snow pea, it's the sweet pea. And this is the type of pea that you don't want to eat. And what this pea contains is a chemical compound called beta-aminopropionitrile that blocks the action of lysyl oxidase and prevents the aldehyde from forming. And without that aldehyde, you can't have cross-linking occurring. So what you can do is you can take extracts from this pea and feed it to our little friend the rat, and then extract tail tendons and test the tail tendons from rats that have been supplemented with the extract from the sweet pea and rats that haven't been supplemented. And as you can see in the plot here, you get a very big difference in the strength of tendon between an animal that hasn't received this supplement and one that has received the supplement, and as a result has not had proper collagen cross-linking in its tendon. Okay, so here's another plot showing the change in mechanics of rat tail tendon. Again, in this case, a lot of things in the tendon literature have been done historically over time using rat tail tendon because it's very easy to acquire, and also the tendons are very nice and long and uniform and simple to work with. Maybe not the best model, as it turns out, for tendon structure function investigations, but different story for another day. So here's the change in mechanical response on what is this, a stress strain plot? Stress strain plot of tail tendons from rats of different ages, and you can see the increase in strength as we go from a rat of a young age up to a rat of 3.5 months old, 10.5 months old, 21 months old, 23 months old, right? So those changes are largely occurring because of changes to the cross-link profile of the tendons over time as the animal ages, right? But the story between cross-linking and tendon mechanics is not so straightforward. Just that more cross-links equals a stronger tendon, right? That would be the logical conclusion, and that's actually the how the thinking was through a lot of decades in the literature. But then people started doing a little bit more work, and we ended up with papers that had results that are sort of encapsulated in this paper's title, right? So the lower strength of the human posterior patellar tendon seems unrelated to collagen cross-linking or fibril morphology, right? So here's some work that I've done and some other groups have done too, to sort of explore a little bit more this relationship between cross-linking and tendon strength. So I've used this really convenient model that's a bovine forelimb model, where within the bovine forelimb there are these two tendons that are very different in terms of their function and the stresses that they see in life. So there's the superficial digital flexor tendon here on the back of the forelimb that really helps power locomotion, and much more so in a horse than a cow, but the stress is still quite high, or supposed to be quite high, from the analysis that's done from cow forelimbs. So the stress in life that this tendon is supposed to see is about 70 megapascals. Now on the front of the forelimb, there's a tendon called the common digital extensor tendon. And this tendon is largely active when the cow's hoof is off the ground, to reposition it in order to land properly on the ground for the next step. So consequently, the tendon operates under a very low amount of stress. Okay. Now it sort of makes intuitive sense, right? That the tendon that's seeing the higher stress in vivo would have more cross-linking. That's indeed what we've found. We meaning both myself and other people within the literature have observed that the superficial digital flexor tendon has more intermolecular cross-linking between collagen molecules than the common digital extensor tendon. But when you look at the strength of these two different tendons, you'd of course assume that the tendon with more cross-linking would be stronger, but we found the complete opposite, that the extensor tendon with less cross-linking is actually stronger than that more highly cross-linked tendon that experiences more stress in life. So this is a really sort of initially an odd result that you might not expect. And this is another plot just from a different group showing you that somebody else has found the same thing as we have. You know, you should never believe the data that's presented by an individual as being, you know, the absolute truth on something. So somebody else has observed this same thing, where the flexor tendon is shown to be stronger than this lower stress extensor tendon. So to explore this a bit more and get an idea of, are the differences in strength between these tendons coming from differences in the strength of the collagen fibrils, or the mechanical properties of the collagen fibrils, or some higher level in the structural hierarchy? One of my master's students did some work looking at the mechanical properties of single collagen fibrils extracted from a tendon. And I'll just take you briefly through what Andrew did here, because it's kind of interesting. So Andrew extracted single collagen fibrils from these flexor and extensor tendons, and you can see one collagen fibril here. And then he glued them onto a glass dish using two epoxy strips, isolating a segment of the fibril in between those two epoxy strips that mechanical experiments could then be done on. And he did his mechanical manipulations using an atomic force microscope, an AFM. So an AFM is generally used to image very small things, and it sort of consists of a cantilever with a a tip on the end, and you go around poking things, and you can, for example, use it so that every time you poke the tip down, you have a certain maximum force set point that you achieve, and you measure the position of the cantilever where you get that maximum force reach. So you can map the height of very small things using this technique. Now, Andrew used this atomic force microscope in a way that it was not designed to be used. So he used it as a very small mechanical loading device, where he'd take the tip of the cantilever, press it down onto the glass slide in front of this segment of the fibril held by the epoxy strips, and then shift the slide or the glass substrate laterally so that the tip of the AFM cantilever stretched the fibril out just like a bowstring, like if you were pulling out a bowstring. And he was able to measure the amount of stress that was being applied to the fibril, or the amount of force that was being applied to the fibril, and back calculate stress based on the diameter by measuring how much that cantilever twisted when the fibril contacted it. So here's an actual picture, and it's pretty dim, but maybe you can see here the fibril, which is a thin strip indicated there, and then the AFM tip, which is just a point when you're looking from above. This is before, here in this picture, before when contact was made between the tip and the fibril, and then down here is an image from
I'm having trouble hearing you.
From during stretching, when that fibril is pulled back like a bowstring in tension. And what we were able to do is produce stress-strain plots from this. And so this is a stress-strain plot from one of these fibrils from the low-stress extensor tendon. And you can see you get a sort of a higher slope or a higher stiffness region of the fibril that then sort of levels off into a lower stiffness region of the fibril. Now, in terms of the mechanisms that we talked about last lecture, this initial high stiffness region can be attributed to the straightening out of those molecular kinks that you can measure by X-ray diffraction. And then the lower modulus region would correspond to when collagen molecules start sliding relative to one another. Okay. So when you rupture one of these fibrils, you get that very deformed look that's very similar to what we saw in the electron micrographs from yesterday, where you've got the fibril with all of these kinks occurring along it. Now, the fibrils from the high stress tendon behaved very differently, where this portion of the curve that's associated with molecular sliding was very short, and there seemed to be a limit to the amount of molecular sliding that would occur. And that's you can understand that if the fibrils got more cross-linking between collagen molecules, then there can probably be less sliding between the molecules before those cross-links are really engaged and start resisting that sliding strongly. And at that point, you get an increase in modulus. And when you look at a broken fibril from these energy storing tendons, right, there's no apparent plastic deformation of the fibril at all. So it looks like it has remained structurally intact until it has brittly fractured at some location. So the story seems to be that even though these tendons that undergo high stress and store energy to help power locomotion are less strong than the lower stress tendons, they have fibrils that are much more resistant to structural damage, right? Or structural rearrangement of their substructures. And then this comes into play a bit when we start talking about damage created by repeated loading of structures. So when you think about an engineering material or a material that's going to be subjected to loading, you want to not only think about how strong a material is, but how resistant to damage a material is as the result of repeated loading cycles. Okay. So let's think about just briefly how many loading cycles something like your Achilles tendon might undergo in a year or in a decade, right? So just as an example, for myself again, using me as the example here, we already said from the last slide that I showed where it's got sort of my running cadence on, so how many steps per minute I generally take when I run. So I do usually 163 steps per minute, and of course that is the number of times the feet hit the ground in a minute. So the number of times a single foot hits the ground in a minute would be 1/2 of that amount. And then if we multiply that by, let's say, the total time that I'd run in a year, which last year was 25 hours. Here we've got 25 hours of running, let's say, multiplied by 1 hour is 60 minutes. That gives us the total number of loading cycles that one of my Achilles tendons would have gone through or experienced in a year. So let's just check out what that number is. So we've got 163 divided by 2 multiplied by 25 multiplied by 60. We've got 122,250 cycles per year, right? So per decade then, so for every 2 years, or for every 10 years, that's 1.2 million cycles for 10 years, right? So it's a fair number of loading cycles that that a tendon's experiencing. So let's look at how materials tend to respond to repeated loading. So as you load, so a conventional engineering material, right, like metals, for example. Here in this case, I'm continuing along the theme of the metal alloys that implants are often made of. So we've got titanium, aluminum, vanadium in this plot. And what you've got in this plot is the strength of titanium, aluminum, vanadium on the vertical axis, and on the horizontal axis you've got number of loading cycles. And loading for these experiments is generally done in sort of a regime that's typically like this, where you've got time on this axis and you've got stress on this axis, and you have some sort of sinusoidally varying stress, where you've got a maximum stress up here that's maybe tensile, and a minimum stress down here that's maybe a compressive stress, and some sort of average stress here. So you take a material and you put it through this cyclic loading, applying tension, compression, tension, compression, tension, compression. Here's one cycle. And you put a material through a different number of loading cycles, and progressively during loading you stop and you break a piece of the material, find out its strength after a certain number of loading cycles, and plot that, and you get one of these what are known as SN curves strength number of cycles. And you can see that the strength of the metal drops off pretty rapidly with increasing number of cycles, right? So the strength of titanium, aluminum, vanadium when it's freshly manufactured is around 1,000 megapascals, and by let's see, what, 1,000,000 cycles down here, we're at around 550 megapascals. So it's lost about half of its strength in 1,000,000 loading cycles. So think about that in terms of your tendon now, right? In 10 years, you've got a 1,000,000 loading cycles. You can't have a tendon that's losing half of its strength in undergoing that many cycles. And of course, a biological tissue is much different than a conventional material, because you have the potential for the body to be repairing damage that's accrued. So instead of an SN curve as shown for the titanium alloy, right? Here's number of cycles and stress. You might expect something for, a tissue like tendon to have a curve that's something like the following, where with increasing loading cycles, you get some material degradation, but you also have some repair going on. Maybe not a straight vertical line like that, but maybe, something like that, where you get some repair going on, and then some more damage, and then some more repair, and some more damage, and some more repair, so that the stress that you see, the peak stress that you see when you're doing something like running, if that's 100 megapascals, you're always above that, right? So that would be a very sensible thing to have happen for something like an Achilles tendon. Now, the really interesting thing is that it doesn't look like this happens at all with your Achilles tendon, actually. So this is some data from what's known as a bomb pulse experiment, or a bomb pulse study. So in the late 50s, between 1955 and 1963, there was a lot of nuclear testing that was done. And as a result of that nuclear testing, there was a huge increase in the amount of carbon-14 in the atmosphere. So that's carbon with 8 neutrons rather than 6 neutrons. So on this plot here, you've got carbon-14 concentration, and you've got date on the bottom, right? And the solid line here shows the atmospheric level of carbon-14 over time. So you can see that it was quite low, and then the onset of nuclear testing, it spiked way up, and then in 1963 there was a large reduction in the amount of nuclear testing that happened. So it's been slowly returning back to the normal baseline level ever since then, right? The carbon-14 that's in the atmosphere ends up being incorporated into all living things, right? It's brought into plants, and then animals eat plants, and then it becomes part of your tissues, right? All of our tissues. And what the authors in this study did was they looked at the amount of carbon-14, the concentration of carbon-14 in some different tissues. So the study, or the sampling date, was when they actually sampled the tissues, was in 2000. Okay, all of the dots on here represent the birth year of individuals that tissue was taken from. Square dots, tissue taken from the Achilles tendon. Round dots, tissue taken from skeletal muscle. Now, if you have a tissue where the collagen is being turned over frequently, or where the tissue is being turned over frequently, you would expect that the carbon-14 level in the tissue at the time of sampling would match the atmospheric concentration of carbon-14 at the time of sampling. And this is indeed what's found for skeletal muscle. So you can see here the carbon-14 concentration at the time of sampling, right? And regardless of birth year of the individual, right, the amount of carbon-14 in skeletal muscle was the same as the atmospheric concentration of carbon-14, right? So indicating that that tissue is being continuously turned over. Okay, but that's not at all what was found for the Achilles tendon, right? So in a lot of cases, the amount of carbon-14 in the Achilles tendon at the time of sampling was far greater than the concentration of carbon-14 in the atmosphere at the time of sampling, indicating that carbon-14 had been incorporated into the tendon at an earlier time and then hadn't been turned over. So it had remained there in place. And you get, there doesn't seem to be in this plot a whole lot of correspondence, right, between these rectangular, these square dots in the actual line that's indicating atmospheric carbon-14. But if you do a 17-year average on that line, then you get an actually really nice correspondence between those plots and the line. So this dashed line here is the 17-year average. So each point on the line is the average of the 17 years following that time on the x-axis. And so what this indicates is that the collagen that is incorporated into your Achilles tendon during the growth period, so the first 17 years of your life, stays with you for the rest of your life and isn't turned over. So the collagen that's in your Achilles tendon now is going to be with you for the remainder of your life. And that's really interesting when we come back and think about this and the potential loss of strength due to cyclic loading of a tendon. And here, what looks like an actual inability to repair and replace damage. Okay. So it seems like a compromise has been made. So the lower strength of the fibrils or the lower strength of energy storing tendon, the tendons that undergo high stress, is in some way related to those fibrils having, or the consequence of those fibrils having a structure that is very resistant to damage as the result of cyclic loading, which is important because those fibrils are not being turned over. All right. Of course, things don't always go well, right? Even though there might not be a great ability in these tendons to remodel and repair damage, and the fibrils might be quite resistant to damage. Sometimes damage does occur, and this is seen a lot in tendinopathy. Now, tendinopathy used to be called tendinitis, and itis being something that's associated with inflammation. And then there was a big debate and a lot of research, and people said, no, there's no inflammation in tendinopathy. We should call it tendinosis. General osis being a general degeneration of structure without inflammation. And then later, after it had been called tendinosis for a while, people said, "Oh no, but there is some inflammation." And then the question of, "Whoa, what exactly is inflammation?" came up. So, in the end, it was settled that it should just be called tendinopathy, right? So this is the more current term. If you hear somebody say tendinitis, tendinopathy is now the preferred terminology for pain and loss of function in a tendon attributable to an overuse injury or an associated with an overuse injury. tendinopathies happen in lots of different tendons commonly. So you get a Achilles tendinopathy commonly, patellar tendinopathy, right below your kneecap there. Lateral and medial epicondylitis, and you notice that still has the itis on. It's actually now elbow tendinopathy instead. Lateral epicondylitis is more commonly referred to as tennis elbow, where medial epicondylitis is called golfer's elbow. Patellar tendinopathy, often referred to as jumper's knee. So you can see where the understanding that this pathology is related to overuse comes in with all of these references to sport, because they much more commonly happen to people who are involved in these activities with highly repetitious and forceful activities involved in them. Rotator cuff in your shoulder, also something that frequently sees tendinopathy. So let's look at the structural change in tendinopathy, or some of the structural changes. So here's a micrograph of normal collagen, and you can see like little black elongated dots throughout the structure. Those are the tenocytes, so the tendon cells that are living amongst the collagen fibrils. And here's one of the things that is frequently seen with tendinopathy. So an increase in the number of cells present, and also a rounding of those cells. So cells generally, or at least fibroblastic type cells, the cells that make collagen, tend to take on a morphology that is indicative of the type of stress being experienced in the tissue. So where you've got tendon and it's largely under uniaxial tension, the cells tend to elongate along the direction of tension. Whereas in articular cartilage, where you've got hydrostatic pressure, so force on all sides, cells tend to take on a more rounded morphology, and we'd call the fibroblasts that are in cartilage chondrocytes. So the rounding of the cells indicates that there's an abnormal mechanical environment sort of being experienced here. You also tend to see neovascularization, or the ingrowth of new blood vessels into tendon, and those are shown down here in those sort of hypercellular areas with lots of cells and sort of a rounded morphology there. And you see a general breakdown of the collagen structure as well. And this is all at the micro scale. Some people have also looked at changes in tendinopathy at the nanoscale. So here is the nanostructural picture of collagen fibrils in a normal tendon. So you can see, again, those fibrils having that striated debanded appearance, and the fibrils being more or less parallel to one another. And here's a tendon that is that has some tendinopathy, and you can see a real general lack of a nice structural arrangement of those fibrils, and some kinking of the fibrils, and etc. People have tried to quantify a little bit the changes at the fibril level that's seen with tendinopathy. So here is a plot showing the diameter of collagen fibrils and the diameter of collagen fibrils, on the horizontal axis, and the count frequency with which you find fibrils of different diameter on the y-axis, so a histogram. And the normal tendon is in black bars here, and you can see sort of a most common diameter that you'd see for collagen fibrils, and then a trail off. So we've got some bigger fibrils, some smaller fibrils, but overall fibrils that are around sort of the 50 nanometer diameter. You can see in tendinopathy that there's actually more smaller fibrils that are found. People have also looked at other changes in tendons that are occurring in tendinopathy. So this is changes in mRNA for different proteins in tendinopathic tissue versus healthy tissue, showing an increase in things like collagen 1 mRNA, collagen 3 mRNA, and various other proteins there, and some enzymes that are degradative for collagen. So some proteases and inhibitors of proteases too. So there's been various studies looking at the changes to matrix. Okay, so almost through this tendinopathy piece, and then we'll turn it over to questions there. So just a couple of slides left here. So here, this is a paper just looking at some of the risk factors for tendinopathy, and what's really noticeable is the largest risk factor, which is reflective of the common name for all of these in being related to physical activities. Here we've got odds ratio for somebody having tendinopathy, and the greatest predictor is having forceful and repetitive movements, right? Here's another paper looking at the occurrence of tendinopathy in athletes or active individuals versus control subjects who are paired for occupation and age. And the notable thing that this study found is that 42% of middle and long distance runners experience tendinopathy, and only 3% of the general population that's matched for age and occupation do. So the really interesting thing, I think, related to the previous structural slides that I've showed you, and the talk about how tendon nanostructure relates to strength and damage resistance is why 42% of people experience tendinopathy, but the other 58% of people don't. Right? So there's something going on here with tendon structure on one hand, biological process here, and physical loading that hasn't yet been resolved, how these three things interact to determine which individuals are going to experience tendinopathy or are prone to developing tendinopathy, and which aren't. Is there something inherent about the structure of tendons in some individuals that make them susceptible to tendinopathy? Are there deficiencies in biological process that maybe feed into that, and how do these things interact with physical loading? So I'm gonna cut things off there. We didn't get to the bit on spine, but that's okay. Hopefully you found the bit on tendinopathy and cross-linking interesting. And please do ask some questions.